Hi, I'm Blake Clement,
I'm April.
and today we're going to be talking about the chemotaxis of E. coli using three solutions: glucose, fructose, and lactose. So what is chemotaxis? Chemotaxis is the study of a motile cell or an organism, or part of one, in a direction corresponding to a gradient of increasing or decreasing concentration of a particular substance.
Some things to know about the sugars that we're using. So a monosaccharide is made up of one sugar molecule, making it energy efficient during metabolism. However, a disaccharide is made up of two sugar molecules rather than one, making it less energy efficient during metabolism.
Our abstract. So in our abstract, we stated that a bacterium can use a number of molecules for food or energy, but we chose three. We're using glucose, fructose, and lactose, all in 20% concentrations. Through our experimentation, we're going to test which sugar the bacteria preferred best for consumption. When we tested our hypothesis, we came to the conclusion that it was valid.
In our introduction, we mentioned that in our experiment, we will be viewing and quantifying the chemotaxis of the bacteria E. coli. We will be testing a solution of 20% glucose, 20% fructose, and 20% lactose to study their effect on the chemotaxis of E. coli to determine if glucose is a better attractant.
Okay, so our hypothesis stated that the glucose will be preferred because it's a simple sugar molecule, and it's going to require less energy to be taken in or broken down by the bacteria E. coli.
In preparation for the experiment, we will fill 6 test tubes with 5 milliliters of sterile water. We will then remove 0.1 milliliters of water from 3 of the test tubes and replace it with 0.1 milliliters of E. coli to create a 1 to 50 bacteria solution. The solutions will be labeled to keep separate for each attractant being tested.
Controls. So to create our controls, first we need to add 0.1 milliliters of sterile water to 2 petri dishes to make our label, our negative control. We'll then label each of the containers as made ready. After that, we need to make sure that the spreader is cleaned with 70% alcohol and fire before, after, and in between each use. For our positive control, we need to take 0.1 milliliters of our premixed 1 to 50 E. coli solution and also add it to 2 additional petri dishes per labeled test tube. Again, we need to label the containers to keep them separate.
In our methods, we decided that once the controls were completed, that we will fill a capillary tube with 20% glucose solution. Wipe excess solution from the capillary tube with provided materials, and then submerge it in one of the pre-mixed E. coli solutions for 30 seconds. Then we'll wipe the capillary tube with the provided materials. Then we'll place the capillary tube into one of the test tubes with 5 milliliters of water. 0.1 milliliters of the solution will be added and spread across to 5 separate petri dishes. The spreader will be cleaned with 70% alcohol and fired before, after, and in between uses. The petri dishes will be labeled and set aside. The 2 test tubes used will be discarded as well as the capillary tube after this is done.
Moving forward, we'll repeat the same methods described for each the lactose and the fructose solution just to determine if the glucose is the better attractant to the E. coli bacteria. Once all the petri dishes are prepared and labeled, you need to incubate them along with the controls at 37 degrees Celsius. Incubation time will be recorded at the time of the completion. Once finished incubating, we will observe results by counting bacterial colonies amongst our samples and controls. We will then record our findings. After that, you need to run the population standard deviation check and record your findings. We will then repeat the experiment to substantiate our findings. Once again, record our incubation times, bacterial colony count, and the standard deviation of the second and third experiment.
So for our results, our first run of the experiment became disposable once we saw the results. The Petri dishes were cross-contaminated. Therefore, we couldn't acquire any usable or successful data to infer any possible solution for any of the bacteria and sugars.
For the second run, we noticed that the glucose solution contained a number of bacteria, housing growth between 100 to 200 colonies. However, the fructose solution supported less growth than that, only containing about 50 to 100 colonies. Last but not least, the fructose solution seemed to house less than the others by a great quantity. The bacteria only seemed to range from about 25 to 40 supported colonies. The results that we saw were agreeing with our hypothesis so far.
In our third run, our results of the experiment, we noticed the same occurring frequency of results. Our glucose solution inhabited the same average of colonies per Petri dish. The fructose solution also reflected the same data. However, with slight variation to its previous average, it still proved to be less than attractant than the glucose solution. In the Petri dishes containing the lactose solution, we noticed a slight and subtle change of colonies. The average varied by only a few bacterial colonies, but not enough to prove itself any more reliable than in the previous one. Therefore, we acknowledge lactose as the least preferred sugar of the three: lactose, fructose, and glucose.
Conclusion. So, in our conclusion, the results of our experiment follow what we stated in our hypothesis. Glucose portrayed itself as the most popular and reliable attractant. The other samples contained fewer colonies of bacteria, which one could infer that E. coli wouldn't prefer the given solution. The positive correlation of number of colonies with the simpler sugar helped us conclude that the bacterium preferred the less complex sugars or monosaccharides over the more complex sugars or disaccharides.
In our discussion, we focused on mentioning that after noticing the results between the solutions, we came to infer that the lactose was less preferred because it is a disaccharide. However, glucose and fructose are only made up of one sugar molecule, making the completion for the first choice logical. This aligns with our results, reassuring us that our experiments concluded in a reasonable outcome, meaning that glucose was the more popular attractant.
Future improvements. So, our experiment could have been improved in the future by better cross-contamination prevention because we lost the whole trial due to lack of a sterile environment. The main focus for sterilization was the exposure to the local air for too long. In the future, one must return the cap of the solution immediately after taking the sample instead of placing it on the table. As far as sterilization of the spreader goes, the groups perhaps didn't let all the ethanol burn off the tool before using it, hindering the results of the experiment. These aseptic techniques are a must for providing not only a positive but reliable result. The second and third runs were better, but with more positive results. We could have had more accurate and more quantified readings.
And these are our references. So if you are interested in looking at any of the journals that we looked at for our experiment and our paper and everything that we did with the chemotaxis experiment, we do have a list of the ones that we use right here. There are also more online, and there are also more publications from these authors and from these websites.
Okay, bye.
Bye.